So today we're going to be having an overview of cytokines. What are cytokines? Cytokines are small secreted proteins or glycoproteins that act as messengers in the immune system, produced by many cell types such as immune cells like macrophages, T cells, or non-immune cells like endothelial cells and fibroblasts. They bind to specific receptors on target cells to influence cell behavior, such as activation, differentiation, proliferation, migration, survival, or apoptosis. That's programmed cell death. What are the major categories of cytokines and their general roles? We have interleukins, which are mostly from leukocytes. They regulate immune responses, inflammation, and cell growth. We have interferons. They are especially important in antiviral responses, modulation of immune reactions. We have tumor necrosis factor, which carry out the role of inflammation, apoptosis, and immune regulation. We have colony stimulating factors, which control production and differentiation of blood cells in bone marrow. We have transforming growth factor, beta family, often immunosuppressive, regulates repair and fibrosis. We have chemokines, which are specialized in directing migration, that's the chemotaxis of immune cells. So in the next slide, we are going to be looking at some key cytokines and what they do. So here we have interleukin 1, which is subdivided into interleukin 1A and interleukin 1B. The main source is from macrophages, monocytes, dendritic cells, other antigen presenting cells. What is its major function? They promote inflammation, induce fever, they are actually pyrogens, activates lymphocytes, stimulates acute phase responses, aids proliferation and differentiation of immune cells. We have interleukin 2. The major source is T helper type 1 cells, activated T cells, and NK cells. Its main function is it stimulates T cell proliferation, enhances natural killer cell activity, supports B cells, general growth and survival of T lymphocytes. We have interleukin 3, which is secreted by activated T cells, supports growth and differentiation of hematopoietic progenitor cells. We have interleukin 4, which is secreted from type 2 helper T cells and mast cells as well as basophils. They promote differentiation of naive helper T cells into Th2 and stimulates B cells. We have interleukin 5, which is secreted by Th2 cells and mast cells. They aid eosinophil growth and activation, support B cell differentiation. They also carry out the function of class switch to certain antibody types. We have interleukin 6, which is secreted by macrophages, T helper cells, fibroblasts, and many others. They carry out pro-inflammatory actions. They stimulate acute phase response, such as B cell maturation into plasma cells, T cell activation, sometimes anti-inflammatory effect via interleukin 10 or interleukin 1RA pathways. We have interleukin 7, which is secreted by the bone marrow and thymic stroma cells and epithelial cells. They support survival and proliferation of lymphoid progenitor cells. They are important in T and B cell development. We have interleukin 8, which is secreted by macrophages, epithelial cells, and endothelial cells. They carry out the function of chemoattractant for neutrophils and some T cells. Plays a role in acute inflammation, also in angiogenesis. We have interleukin 10, which is secreted by T cells, especially regulatory T cells. B cells, and macrophages. They are broadly involved in anti-inflammatory actions. They inhibit pro-inflammatory cytokine production such as interleukin-1, tumor necrosis factor alpha, and interleukin-12. They down-regulate antigen presentation. They also support B cells. We have interleukin-12, which is secreted by macrophages and dendritic cells. They carry out the function of T cells, especially regulatory T cells, B cells, and macrophages. We have interleukin-17, which is secreted by T helper cells 17 type, other immune cells. They have a potent pro-inflammatory action, induces other cytokines or chemokines such as interleukin-6, GCSF, tumor necrosis factor, etc. They are important in defense against extracellular pathogens involved in autoimmune inflammation. We have tumor necrosis factor alpha. They are secreted by activated macrophages slash lymphocytes. They carry out the role of major mediator of inflammation, can cause apoptosis, stimulates acute phase reactions such as fever, helps in recruiting immune cells. This regulation can lead to pathology, e.g. septic shock. We have interferon gamma, which is secreted by Th1 cells, natural killer cell, and NKT cells. They carry out the role of activation of macrophages, support class 1 and class 2 major histocompatibility complex expression, enhances antigen presentation, supports cell-mediated immunity, such as antiviral effects. We have transforming growth factor beta. They have many sources such as T cells, macrophages, and some non-immune cells. Generally are anti-inflammatory, inhibits proliferation of many immune cells, promotes regulatory T cell development, involved in tissue repair and fibrosis. We have granulocyte macrophage colony stimulating factor, which is secreted by T cells, macrophages, fibroblasts, and endothelium. They stimulate growth and differentiation of granulocytes and macrophages from bone marrow precursors. They also activate these cells. We have granulocyte colony stimulating factor, which are secreted by fibroblasts, endothelium, macrophages, etc. They promote neutrophil production and function, used clinically to increase neutrophil count. We have pro versus anti-inflammatory balance. Some cytokines promote inflammation, that's the pro-inflammatory action, such as interleukin-1, interleukin-6, interleukin-8, interleukin-12, interleukin-17, tumor necrosis factor alpha, interferon gamma, etc. Others suppresses or regulates the inflammatory response. We have interleukin-10, TGF beta, possibly interleukin-4 in some contexts. Thanks for listening.